I will try to keep you awake on your delicious lunch and explain what I'm doing here, talking about oyster genetics, which as I received in an email from Jim Bristow in January of 2008 was he identified oysters as on the edge of the envelope of what JGI does. And at that time, there was a lot of pressure from OMB for JGI to get away from these various projects they'd engaged in, which had great evolutionary interest, and really, stick to the business of energy. And I could appreciate that. And so the first part of this talk is kind of a long version of what I answered him, what the rationale is between oysters and energy. And the last part of it is basically a report on a project that we've just finished this last year. EST, large-scale sequencing project, ESTs, and some BACs. And I want to call attention to my co-authors, Andy Gracie and from USC, and Pat Gaffney from the University of Delaware, who helped and are helping, continue to help analyze these sequence data that we receive from the JGI. So what do oysters have to do with energy? Well, first of all, here's the critter. And it takes a French poet to say something like this. Of course, they're a food item. And I have to confess that my interest in them is as a food item, but they do have a role in energy. We could talk about, for example, the oyster I work on, the Pacific oyster, has the highest aquatic production of any organism on the planet. About 4.42 million metric tons per year are harvested. This organism is grown on all continents but Antarctica, so there's a strong global interest in it. And we could talk about, for example, the calcium, the carbonate, the CO2 that's taken up by the calcium carbonate shell, but we wouldn't want to make a big deal of that, and I'm not really going to say much about it. The annual harvest of the Pacific oyster is about 6/7 of the weight is the calcium carbonate shell, which means that the annual harvest of the Pacific oyster sucks up the carbon dioxide equivalent to the output of the country of Cameroon, which is pretty far down the list. But that's just this species. So clearly these higher organisms that do sequester carbon in the form of calcium carbonate and aragonite are playing a big role in the global biogeochemical processes. And as I said, I'm not really going to talk much about that. I'm going to focus instead on the growth of the animal, the production characteristic, and it shares. The oyster shares something with plants that I think makes the argument that this has something to do with energy. And what they share is this phenomenon of hybrid vigor, shown more than 100 years ago now by George Harrison Shull. Corn is a naturally outcrossing plant, but when you self-fertilize it by detasseling the plant and controlling the fertilization and then cross those inbred lines with each other, you get hybrid corn, which is the basis of production of corn in the United States, and we've had a 7-fold increase in the production of corn in the United States as a result of this phenomenon, empirically applied. So they simply make inbred lines constantly every year, thousands of them, test them in all possible combinations, millions of combinations, grow them on millions of plots throughout the United States to determine the best combinations, and then take those parent lines and produce them. And this is almost a slope of 2 bushels. This very metric measurement of corn production, bushels per acre. It's a big basket of ears. And you can see that George Harrison Shull came along here in 1908, so it took several decades for this phenomenon to be incorporated into commercial breeding practices. But once it was, it's taken off. So I became aware of the fact that the poplar genome that was sequenced here was a hybrid clone. Corn, sorghum, all these plants, they're improved by this phenomenon of hybrid vigor, and yet we know nothing about how that actually works. It's an empirical practice. We don't really know the biology underneath hybrid vigor. What causes this organism to grow bigger? That's unknown, still unknown at present. So oysters have the same phenomenon. This is a picture of a cross between 2 inbred lines of oysters. Again, oyster is naturally outcrossing species, broadcasting sperm and eggs into the water. Fertilization is external. The babies float around in planktonic form for 2 or 3 weeks and then settle out. And we showed early on by making inbred lines out of these that we got the same kind of phenomenal increase in the growth rate of the hybrids. So this is yield. Yield is a component phenotype that's part growth and part survival, and both parts are involved here. They have greater survival and greater growth. So now that we have this kind of plant-like biology in an animal, there's all kinds of neat things you can do with animals that are much more difficult to do with plants, namely physiology. But before I get to that, first I want to convince you that this is not just a freak cross that happened once, but we see this virtually every time we make inbred lines and cross them. These are the results of a cross of 5 inbred lines and a so-called diallel cross, that is all 5 lines used as both male and female parents, producing 25 families. And this is seed weight, so these guys were harvested, look at a point here, at a rather small size, each animal about 3 grams. The boxes are the parents, so on either end of the box is where the parent line is. So you can see the 92 parent is that far line, and every time there's a 92 here, you see that far line. 92 inbred oysters were about 3 3/4 grams in weight. And so the thing that will jump out at you is that all the parents, all the boxes line up over here between 2 3/4 and 3 3/4 grams. All the hybrids, the circles on this graph, are the hybrid combinations between them, and you can see in every case the hybrid is bigger than the best parent. And the dark filled in circles, they're significantly better than the best parent. The error term here is a cage of oysters, so we only have a few cages. The errors are quite large in these experiments. Nevertheless, it's pretty obvious that hybrid vigor is very pervasive, very repeatable. Almost any time you make an inbred line out of this outcrossing population, you can get this phenomenon. And so this has set up a program where we can begin to look at the genetics and the physiology of this. And one question I always get, well, how do you know those are better than wild oysters? And that's work in progress. So this is a graph showing the comparative yield. Again, this is kind of seed-size oysters, 100 of them for different crosses compared to wild, the current stock grown by the industry, wild oysters from Willapa Bay in Washington, the inbred lines, and some various crosses. And so we have some hybrids that are producing 2 or 3 times the yield of wild oysters, and we even have some inbred lines that are producing better than some of the wild oysters. So clearly this is going to be an important basis for improving aquaculture species. This is the fastest growing sector of food production. Oh, I'm not supposed to talk about food production, sorry. Anyway, this will have practical application, but what we're really interested in are the causes. And the causes of this phenomenon have actually been, again, this has been a century-old puzzle. Where people have been trying to understand this both at a genetic level and a physiological level, and we're still not there yet. But here are the genetic causes that have been around for a century, still not resolved. The easiest way to think about this is that yield is governed by some important genes, and at these genes, the heterozygote genotype is somehow superior. We won't talk about the physiology of that, but if that were the case, then it's quite easy to see why hybrids are outproducing inbreds. It's been very difficult to actually show this mechanism, however. And that's been primarily because there's an alternative mechanism to explain it, and that's dominance, where the heterozygous genotype is, I might be running out of, is no better than the best inbred parent line. Now, how can that cause hybrid vigor? That's clearly not hybrid vigor. You got the hybrid being as good as the next parent line. What comes about here is that the character yield is governed by hundreds of thousands of genes, and so you have the multiplication of this phenomenon across all the underlying loci. At the next locus, the best parent might be the alternative inbred line, and if across all the genes underlying this trait, if you average across all of those, the hybrid is always better, as good as the best parent, then it will come out on average looking superior to either of the inbred parent lines, even though at no locus is the heterozygote superior. So this is the major competing hypothesis, that it's all caused by dominance. And it's been really impossible to resolve the overdominance-dominance controversy. A lot of people think that probably the explanation that people seem to accept now is that it's largely dominance. But we're still kind of looking for the overdominant characters. There are a few other ideas that have been put forward. One is that it's epistasis, that the action is all in the interaction of genes and combinations. That being a more complicated hypothesis has been even more complicated to test. And finally, in plants at least, there's the idea of gene dosage. It's been shown in corn that a triploid corn plant, ABC, putting 3 genomes together is better than AB or AC or BC, and that a tetraploid plant, ABCD, is even better. So you get the compounding of heterosis phenomenon in a gene dosage sort of way. Another thing that's emerged from the sequencing of corn genomes is that different inbred lines, these elite inbred lines in corn, actually have different sets of genes that are not present in the other inbred line. And so a hybrid kind of enjoys this greater gene expression as, by virtue of putting together these very disparate genomes. And to me, that's just kind of the ultimate dominance. I mean, here, inbred lines missing a gene is kind of the ultimate recessive allele. So formally and logically, I would put that into the dominance category rather than the gene dosage category. All right, so those are the competing explanations that have been around for a century, been very difficult to resolve. We're trying to do this in oysters. So we've extracted from oyster populations such as this one and up in the Pacific Northwest and Puget Sound area. These are kind of the natural beds that oysters form. And if you're a biologist and you want to know what makes these critters grow at different rates, it's impossible to really find that out by going out onto this bed and trying to understand this. So we've made these inbred lines derived from these natural populations in the Pacific Northwest. These inbred lines are all from conspecific populations, by the way. They're not different races or geographic subpopulations or different species. They are from the same randomly mating population. We've made the inbreds and we've moved on to the F2. And in the F2 population where you get the segregation at each marker in the genome, it's possible to follow a genomic set of molecular markers and to do quantitative trait loci mapping, QTL mapping, and to try to find out where the genes are that cause this variation in growth rate. And these show 3 different families that were individually tagged. You might be able to see a tiny little tag there if my hand weren't shaking so badly. On each of these oysters, we followed them through, so we have excellent phenotypic data on their growth rates, and so we are engaged in this now common process of QTL mapping. And here's a genome scan down one of these F2 families, about 376 individuals. And the genome is about 820 megabase pairs, roughly 1,000 centimorgans. We have about 700 covered here. If you can see those little red tick marks, there's about 55 microsatellite markers that we've used in this F2 family. And you can see 5 peaks scanning down the genome where the y-axis is a measure of the statistical significance of the relationship between genotypes at these positions in the genome and the association with size. And this is live weight at 10 months. It's just one of the live weight measures. And so, great, we've got QTL. So the real question is, how are the QTL acting? Is it dominance? Is it overdominance? And it's neither one. And I just have recently gotten this result, but basically what we see in every case, the heterozygote is not like the superior parent in its yield. It's like the inferior parent. So it's neither dominance nor, well, it's dominance for the recessive allele. So it's kind of negative dominance. So this is our result. I don't know what it means myself. I guess it has to imply some kind of epistasis, but I'm hard-pressed to find an epistatic model that will give you this kind of marginal underdominance at each case, and yet some kind of global heterozygote advantage. So curious result, but we're obviously need to do more of this sort of work, but we can, through these kind of crosses, get at the genetic basis, and I think the genetic basis is going to be quite interesting. It's obviously not fitting any of the classical ideas, and I think this is a point where you can actually learn when all of your ideas are blown out of the water. So that's kind of the genetic components. What I'm studying, and my colleague Donald Manhan, with whom I've collaborated for nearly 20 years, is more interested in the physiological causes. And there have been some ideas floating around for quite a while. One is kind of biochemical flexibility, that in some range of environmental measure of the environment, this might be pH, it might be temperature, the different genotypes have different local optima, and the heterozygote inheriting an allele from each of those parents might be more broadly adapted across that range of environmental characters. So that's a kind of a flexibility idea. There's an efficiency idea. The fact that for a given set of enzyme activity, the heterozygote might be actually producing more product might be using more of that enzymatic capacity. And that could occur, for example, in these multimeric complexes, multimeric proteins, for example, where the heterozygote can now build a better protein based on the subunits coming from the different inbred lines. And Donald Manahan has seen this sort of thing in the sodium-potassium ATPase transporter, for example. Another idea is kind of a molecular balance idea. Recently, as we've been acquiring information on the expression levels of different genes, we've seen this, and this was seen by a student of Donald Manahan's, Eli Meyer, who's here in the audience, who's now moved on to the University of Texas. And what Eli saw in his dissertation was that you could make a correlation, begin to make a correlation. This is very preliminary data, just 3 points here, but you could begin to make a correlation between the variance in the expression of the genes. These are ribosomal proteins, and yield. So a high-yielding heterozygote has much less variance in the expression of the different proteins that have to go together into this complex molecular machinery, the ribosome. And so this is a kind of molecular balance idea, that hybrids are somehow more in balance and making things more harmoniously. Donald's analogy to this is if you're running an automobile manufacturing plant and the transmissions are arriving faster than the engines, you're not going to be very efficient at putting the automobiles together. And finally, at an even higher level, kind of the organismal level, there's the standard growth rate equation, balance equation, that we can do with animal physiology, where we can look at questions of acquisition and allocation. So this is the acquisition term. The growth rate is a function of how much energy you consume, how efficient you are at assimilating that, minus the respiration, the costs of the metabolism, and the excretion products. And what we've determined is that in oysters, it's about 50-50, that the hybrids are achieving their advantage over the inbreds. About 50% of that is allocation, acquisition, about 50% of it is the allocation. And here's a little bit of data. This is larval, these are larval oysters, so this is a kind of developmental scale, shell length. And at every age during development, the hybrids in the dark dots are consuming, are clearing more algae out of the water than are the inbreds. So they're acquiring more energy, and they're doing more with it. This is studies of protein synthesis and degradation in these two hybrid and inbred populations. And again, the hybrid, this is also a developmental scale, the bigger they get, the more protein they synthesize, nanograms of protein per larvae per day. So at some point during development, if they're synthesizing 100 nanograms of protein, the hybrid is keeping a high percentage of that. The latest figures is more like 70 to 80% of that protein they keep as body protein, they assimilate, whereas the inbred is only keeping about 20 or 30% of it. So incredible differences in the metabolic efficiencies of these organisms. So we have some insights that we feel are rather new relative to what people know about this, the physiology of this in plants. And we've done some transcriptomic analysis using a now dead technology, MPSS, massively parallel signature sequencing, which kind of morphed into the Illumina sequencing. But that analysis told us that if we compare the expression profiles of inbreds and hybrids, we see that most of the expression is non-additive. So in this category, our gene expression is the average of the gene expression in the two inbred lines that went into this cross. And almost none of the transcripts are in that category. Almost all of them are in these non-additive categories where you get something quite different than the average expression level. And many of them are in this overdominant, overexpressed, that these proteins are overexpressed relative to the mid-parent value, or they're underexpressed. They're expressed like the least expressing parent. So we've again got some insights into this, and that's why I think the oyster is a valuable model system to be looking at heterosis. That was my pitch to Jim, and he didn't cut our program. We were able to finish that. So I'd like to give you very quickly now this kind of update on this community sequencing program that we had with JGI. And I'm, I guess, representing an oyster genome consortium that's international in nature because this oyster is grown on all continents but Antarctica. We have lots of people involved in it from different countries. This is the steering committee that I am privileged to represent. And so our project with JGI had two parts to it. I meant to thank, on my opening slide, all the people at JGI have been very helpful, besides Jim and Carrie with the project management and so forth. Dan Roxar for support and inspiration from the very earliest days. And then the people who did the work, Erica Lindquist, who did the EST sequencing, and Jane Grimwood from the Stanford Human Genome Center, who did the back part of this project. So thanks to all. It's been really great working with JGI. It's produced incredibly valuable information for us. So the first part of the project was to sequence some backs that had probe positive for genes of immunological interest, and to judge from that the level of polymorphism in the genome, because genome centers were becoming aware and sensitive to the fact that the level of polymorphism in genomes was going to cause big problems in the whole shotgun sequencing approaching methodology. And the second part of it was to do a large-scale EST sequencing project, which was cut at one point from 150,000 paired ends to 75,000, again, because of the seeming irrelevance of this oyster to the energy project. But this is still a tremendous resource, and we're very happy to have it. And we fed into that some different larval stages and some adult tissues from 2 inbred lines. So our goal here was to not only do gene discovery, but to also generate some information about SNPs, which we could use. So here's a couple slides about each of these. Here's the back sequences. 58 of them were from the Pacific oyster, Crassosteria gigas. 2 of them were from the eastern oyster, Crassosteria virginica. To make a long story short, these were all the back clones that were sequenced, and we were able to put them into contigs. This is Pat Gaffney's work. He was able to put them into contigs, at least for 6 of these, that we interpret as alleles. So these are allelic back contigs made from these sequences. And the question is, how polymorphic are they? They're incredibly polymorphic. So in coding regions, this is again work in progress. We haven't completed these analyses yet. We just got the sequencing this past year during the summer. But the SNP density in oysters in coding regions is 1 out of 65. So every 65 base pairs. This compares in humans to 1 out of 300 or a few hundred. And in introns, 1 out of 21. So we got enormous SNP densities and indels. So either insertions or deletions. There's an indel in introns every 85 base pairs. So these are incredibly polymorphic genomes. In the intergenic regions, where you can even align the intergenic regions and say that they're the same, we're getting incredible densities of SNPs and indels. And part of these indels are transposable elements. It looks like these genomes, like the genomes of plants, are also riddled with transposable elements of all kinds. They're shown here. Particularly these non-autonomous transposable elements. We've got loads and loads of these things, these mites and pearls, quite common and quite active. So what's shown here under the unique active, these are the 2 alleles differing in the presence or absence of these transposable elements. So they're looking like they're presently active and transposing even now, which could explain a lot about the biology. We're really looking forward to that. Here's the cDNA part, various libraries. The last 2 were done by 454 sequencing, and we generated about 1 million reads there. The rest of them were done on various stages, but the bulk of them come from these adult libraries, which again were made from pooled cDNAs from 2 inbred lines, differentially tagged so we could back out which sequence went with which inbred line in the end. Nice high quality sequences there, 700 base pairs or so. Here's just a flow diagram of the initial assembly of these Sanger reads. We started out with 189, screened through them, stringent assembly. We end up with 5' and 3' ends, about 26,000 clusters. So we started with the 5' ends because we were expected to get better blast hits, and blasted them. This is a kind of sequential blast, first against Swiss prot, and anything that blasts against there is removed from this pathway, and then take the rest of them and blast them again. So we get about half of them blasting to Swiss prot, which is excellent. About half of them, this is amino acid blasts. A small number, about 2,000 to the Ladia genome, which is here at JGI. And then a large number to GIGAS EST database in France, which maybe makes sense because it's the same species. Maybe these are bivalve specific. Maybe they're non-expressed sequences too. So a variety of reasons why that might be. We have taken this information off and went back to the clone library and pulled out the clones that represent these sequences, and which are very long insert sizes, and done some Illumina sequencing on that. And we're presently we're hoping to assemble complete sequences for about 9,000 genes, which is about 40% of all the genes they have. So we're going to be in very good shape in terms of having gene models for the Oyster. And we are presently dealing with how to assemble the Sanger reads and the 454 reads. So this is 145,000 and about 1 million of these guys. That's in progress. And we took these 158,000 clusters. Andy Gracie did this just a few days ago, blasted them against the human RefSeq database and pulled out all the human genes for which we have something resembling that in the Oyster, and took that human gene list and blasted it against the human reactome, which is a database, really cool database showing all the reactions. Each arrow is a reaction in the human genome, and these are pathways. And the gray arrows are the kind of background of the human reactome, and wherever there's a blue or a color superimposed on that, it means that we have in the present database of Oyster ESTs something that resembles that. So we have, in other words, pretty good coverage of the human reactome represented in our libraries. And interestingly enough, we have very high representation in gene expression pathways. These are some of the top hits here. The colors indicate overrepresentation. So we have very good or overrepresentation, given the sizes of the samples, of gene expression pathways, or protein metabolism pathways. And these are the very pathways that we think are behind the heterosis thing. So for once, something is going in our direction, and we hope to be able to use this information to do more detailed studies of the physiology of this heterosis trait. And finally, I have to mention that the genome is being sequenced in China at the Beijing Genome Institute. The funding was obtained by the Institute of Oceanology, Chinese Academy of Science, independently of the Oyster Genome Consortium. And they're using Illumina sequencing, shotgun sequencing. We did convince them to use one of our inbred oysters, 4th generation 51 line, so sharing some genes with the sequences that were obtained here at JGI. They have tremendous coverage at this time, but the contig sizes are very small, and so they've got this genome in 1 million pieces, and we can understand why, because this polymorphism is just killing them. So they're working on this and doing their clever things, and hopefully I wish them success. Finally, we've taken the JGI information and we moved on to a project called GIGASNP, funded by the USDA, to discover and map these SNPs that we tried to back out of the sequences. So here's alignments from the 2 different inbred lines showing these sequences. So if we have multiple sequences from each line supporting a nucleotide difference between them, then we feel that these are real SNPs that will be segregating in our families, and we'll be able to put them on the linkage map. And one of our challenges here, well, the goal of this project is to integrate the genetic, the physical, and the cytological maps to support this whole genome sequencing effort, because they're going to need us, I think, in the final analysis to help put the sequences together. And one of the challenges here is that the genome, the exon sizes of these mollusks, so here's the Laudia exon structure for this particular gene, the exons are really tiny. The average exon size in Laudia is 200 base pairs, 212 base pairs, and it looks like the Oyster has the same thing going on. So we have these tiny little exons, many of which can't even support a SNP assay in the usual manner. So this is, again, another challenge that we have. But finally, we get down to the question of what can genomics do for heterosis? You know, if we're going to really understand this phenomenon, what can genomics, and especially the genomics of oysters, do for us? And this is a larval oyster here. It's about 6 days old. This is the velum structure, this ciliated sheet of tissue that they use to catch single-celled algae in the water column. And basically, we're kind of working off this kind of structure where we've got genetics and physiology tied together with genomics to try to have this interdisciplinary understanding of this phenomenon. And what we want to do here is, using our knowledge of nature and nurture, is to really predict what genotypes are going to make hybrids before we actually have to grow them in the field. So that's really the holy grail for us, is this kind of predictive knowledge and a prediction of what the phenotype is going to be based on sequence information or based on the development of biomarkers for efficient metabolism, for example. So that's the conclusion. I hope I've kept to my time limit. Thank you very much.
And the expectation is a heterozygote to the most efficient. In the recent data you showed, the heterozygote was not the largest, it was the smaller one. Does not this suggest that gigantism is evolutionarily selected against, and that while large might be a successful appetizer, large is an unsuccessful oyster? So that the heterozygote is the best, but the best is small size, not large.
Well, we're not making any evolutionary statement about these results. So these results are the comparison of genotypes made in an experimental cross. But I have to say that this variation that we're observing is present in the natural population. So these inbred lines that we derive, we've simply reorganized the variation that is present in the natural population and reorganized it into inbred lines and crosses that we can try to dissect the components of variance that do occur in the natural population. We haven't created anything new. And what we're seeing is this very non-additive type phenotype where heterozygotes are growing faster. Now, of course, how that plays out in evolutionary timescales, I'm hesitant to speculate about. But those components of variance and those kinds of physiologies exist in natural populations.
We have a question over here.
Oh.
Hi. About the genome assembly, actually, because of the heterogeneity of the two copies of the chromosome is so high, I think that will cause a problem of the annual assembler. So have you think about that to use single sperm to amplify the genome and sequence that? Probably multiple single sperm amplification and combine those things will help those assembly.
Thank you.
When you do QTL scan, you normally ask whether alleles A or B, are different in phenotype. Have you tried a model where you actually specifically ask whether AB gives a different phenotype than A and B? So that might be something interesting to try in the QTL scan, to specifically ask whether the heterozygote is different from A and B, which probably wasn't done in this case.
I'm not sure I understand your question.
So QTL scan, what you normally do, you have at every position, an individual has a certain genotype, and what you just ask, is there a difference whether you have A alleles or B alleles? But what you are asking for is something really quite different from what you normally do in QTL mapping, where you ask for differences between A's and B's. You should specifically ask whether AB is different from AA and BB, which probably has not been done, but if it has, will be good to know.
Okay.
So I work with corn, so I've thought about this a lot. Have you looked at small RNAs at all?
No.
Okay.
Yeah, tell me about them. We'll talk. I mean, this is a community of like half a dozen people really working on this stuff. So it's
So how do you like your oysters?
half shell, raw.
Okay, good.
Is there a good oyster bar in town?
Yeah.